Good evening. I'm Michael Mallinson. I'm the president of the Canadian Spondylitis Association. My usual question at these type of events is there anybody here who has never been in a room full of spondyloarthritics before? Hands up. Oh my goodness, look at that. Okay. Well, this is what I think the Canadian Spondylitis Association is all about. Friendship is born at that moment when one person says to another, "What, you two? I thought I was the only one." And I think we know that by and large our journey has been a long and difficult one to diagnosis. If you were diagnosed in under 2 years, you were very lucky. If you're diagnosed at 7 to 8 years, you're about average. If you're exceeding 10, 15 years, then you're not so lucky at all. But anyway, there are a lot of people in this room who have the same disease you have, and I'm sure there were times when you thought you were the only one, and it's gratifying, I hope, to find that there's others of us and that we care and that you are not alone. I wanted to go over this slide quite quickly, and it points to the relevance of the Canadian Spondylitis Association as well as some other relevances. To the right on the screen is the pharmacological treatment of AS. So you're talking about non-steroid anti-inflammatories going all the way through to biologics or TNF blockers. The left side is the side that I like to concentrate on because education, that's why we're here, and we put on these types of forums in conjunction with SPARC and the Arthritis Society. Exercise is all important. Exercise is medicine, and we're going to have a physiotherapist talking about the importance of therapeutic exercise, but recreational exercise is also important as well. Rehabilitation, patient associations and self-help groups. We are a patient association, the Canadian Spondylitis Association. We're entirely volunteer run, and I would like to ask, we haven't really had a support group in Toronto for quite a few years. I'd like to reactivate that, so if anybody here is interested in becoming involved in a support group here in Toronto, would they see me afterwards? Just come down here and see me after this event is over, and we'll talk about how we can reactivate a support group in that fashion. This is who we are and where to find us. We've been around since 2006. We're a baby of SPARC in many respects. I would like you to become members if you're not already a member of the Canadian Spondylitis Association. The more we are, the bigger our voice. There's a lot of advocacy to do, and indeed just talking to other people and attending forums like this can be therapeutic in itself. So you can find us at www.spondylitis.ca. Our website is in the process of redevelopment, so the current website is a little not too good, a little shaky, but in any event. With membership you get our free quarterly newsletter, and I think without further ado, to keep us on time, I have a great pleasure in welcoming Dr. Zinman and Dr. Haroon as well. Thank you.
Okay, good evening everybody, and thanks Michael for that kind introduction. So, for those of you who don't know me, I work in the spondylitis clinic today with Rob Inman. So together we run the spondylitis clinic once, or occasionally we do it twice a week, and together we have around probably close to 2,000 patients with spondyloarthritis, and specifically ankylosing spondylitis patients. We have probably we have seen almost 1,000 patients over the last several years. We have a special interest on the pathogenesis of ankylosing spondylitis, and we've been working on identifying the functional consequences of the new genes which have been identified to be associated with ankylosing spondylitis. We also are interested in novel therapeutics, so we do have a clinical trial center here where we have several clinical trials running. Today I would like to concentrate on what we know about the pathogenesis of ankylosing spondylitis, and Dr. Inman will be speaking about the latest on therapeutics. Okay, so we all know that genes are quite important in the pathogenesis of ankylosing spondylitis. Now, how do we know that? If we look at families with one member of ankylosing spondylitis, you're likely to see another member with some form of spondyloarthritis or inflammatory bowel disease or psoriasis in the family. So there's a high likelihood of recurrence of diseases in this family among members who are related. So you can do these genetic studies either by looking at twins, where there is a high degree of sharing of genes, or you could look at by studying families. Traditionally, the way to do this is by comparing the genes present in patients and compare those to the prevalence of the same genes in controls. And you can do this in two different ways. Either you can have a previous hypothesis. For example, we know that there's a lot of inflammation, so we might decide to look at specific genes which are driving inflammation, like TNF, or the immune genes like major histocompatibility genes. So we can just focus on these genes, or we could do it in a non-hypothesis way, that is by looking at the entire genome. So this has been possible over the past 5 or 6 years because of the explosion in our knowledge about the human genome. So the Human Genome Project has given us the entire sequence of the human genome, so we could test the entire genome in all the cases versus the controls. So that has really led to a significant increase in our knowledge regarding the pathogenesis of AS. So these are some of the very early studies which were done in ankylosing spondylitis, and the top the table which you see there is the comparison of the concordance of ankylosing spondylitis in monozygotic twin pairs versus dizygotic twin pairs. And as you can imagine, monozygotic twin pairs have greater gene sharing, and if genes are important, you would expect greater concordance in monozygotic twin pairs. And as you can see, 50% concordance in monozygotic twin pairs versus only 20% concordance. So in other words, if you have 10 pairs of twins, and if there are 5 affected members among the 10, you at least 5 out of the 10 would have both twins affected, compared to only say 1 in 5 dizygotic twin pairs. Now, if you look at the table on the bottom, it shows the recurrence risk, or the chance of recurrence of ankylosing spondylitis, and the table shows what the degree of relationship is with the first person who gets the disease. You can see in the first column, monozygotic twins, the recurrence risk is 63%, and as the degree of relationship goes down to siblings, parent-child, first degree, it drops down to around 8%. And as soon as it becomes second degree and third degree, it drops rapidly. So this is again to show that the more genes we share, the greater the likelihood of having a recurrence of ankylosing spondylitis. So clearly, there appears to be a significant role of genes in the pathogenesis. This was one of the earliest studies which identified a gene in ankylosing spondylitis, and you can see the date there. It's 1973. It's more than 40 years ago that we first came to know that this particular gene, HLA-B27, is seen in 96%. These are earlier figures. The figures are slightly lower, probably around 80% now in ankylosing spondylitis, compared to around 4% in controls. So the prevalence of B27 among the Canadian general population is around 7%. So clearly, if the HLA-B27 is positive, the chance of having ankylosing spondylitis is high, but that doesn't mean that everybody will get AS. So I'll come to those figures in a minute. So what do we know about HLA-B27? We keep talking about the association of HLA-B27 with ankylosing spondylitis. As I mentioned, more than 40 years has passed since we identified this association first, and yet we don't know exactly how HLA-B27 causes the disease or how it contributes to the pathogenesis. But based on the known functions of HLA-B27, we could come up with some hypotheses and possible scenarios how this might work. So as you see in the figure, HLA-B27 can be present on the surface of immune cells, and these immune cells have a function to present antigens inside the body. So essentially the immune function of the body is to prevent external antigens or external infections from setting up in the body. So you want to get rid of these infections. So antigens from these external agents or bugs are being presented to our immune system, and the effector arm of our immune system gets rid of it. So maybe HLA-B27 is presenting some form of an antigen on the surface, and our body is reacting to that in an abnormal way. So that is one possible explanation. The other and more interesting explanation is based on the peculiar property of HLA-B27. As you'll see here, those white strings which you're seeing here are HLA-B27 molecules which have misfolded. Or in other words, they don't fold properly, they don't come to the surface, they just accumulate inside the cell. And when a large number of B27 molecules accumulate inside the cell, there is a process of cell stress being stimulated, and significant increase in inflammatory cytokines can happen and be released. So it may not be an immune response at all. It may just be a lot of inflammation being triggered by excess misfolding of HLA-B27. So these are the possibilities. And one last possibility, which is also quite popular, is the fact that these misfolded B27 molecules could come to the surface, and they may be recognized by some other cells which are not supposed to be immune reactive cells because of this abnormal presence of B27 on the surface. So these are the possibilities. Now, is B27 important? If you introduce HLA-B27 into a rat model, almost all the manifestations which are seen in humans can be seen in this rat model. You'll see arthritis developing, psoriasis nail lesions, psoriasis skin lesions, and even inflammation in the bowel developing just by introducing HLA-B27 into the rat. So clearly HLA-B27 appears to be quite important. And as I said, close to 90%, around 85 to 90% of ankylosing spondylitis patients have this gene. But if you consider 100 people with HLA-B27 just from the population, between 2 and 5% only actually develop ankylosing spondylitis. So having the gene doesn't mean the disease is inevitable. So there is an almost 98% chance that the disease will not develop, even though the person is HLA-B27 positive. So clearly it appears that B27 is important, but it may not be sufficient. There are many other players that are involved in disease pathogenesis. So the breakthrough came, as I said, with the whole genome studies. With this small single slide, you can actually test the entire genome with one drop of blood. The patient's DNA and the control DNA is introduced into the slide, and you can read whether the genes, which variant, whether a normal variant or an abnormal variant is present. You compare this between the cases and controls, and you can come up with statistics to identify which genes are involved. Okay, and this is how this particular plot would look like in ankylosing spondylitis. On the y-axis, or you can see here a tall, each of these colors is a different chromosome, and each of these dots which you see refer to different genes in the chromosome. And what you see here is the level of significance of association, and the most important, or the tallest one here, is HLA-B27 as expected. And although it is called a Manhattan plot, Dr. Inman keeps saying it resembles more the Toronto skyline. So it's probably the CN tower of this plot is actually B27 here. The second strongest association that we have seen is with a molecule called ERAP1. It is a very interesting molecule. We have done some work on it. I won't go into the details, but I'll share a few details with you. And then another very interesting gene is the IL-23 receptor, a different cytokine. How many minutes am I in? Okay. So I'll make this very quick in the next couple of minutes. So you can see these are the several genes which have been identified over the past 5 or 6 years. So from HLA-B27, a single gene 40 years ago, now we have almost 20 or 25 genes which have been identified in AS. Now we just have to sort out how they cause ankylosing spondylitis. So here is a simple cartoon to show you how B27 may be interacting with another gene, ERAP1, and causing ankylosing spondylitis. So you have these antigens being generated in the body, and these antigens have to be trimmed to appropriate lengths or shaped so that they can sit on HLA-B27 and they are presented to the surface. So if this happens in a normal manner, you get a normal immune response. But you have this molecule here, ERAP1, which is responsible for this shaping function or trimming function. And as you saw in the previous graph, this is probably the second most important gene in ankylosing spondylitis. Now, if there is something abnormal with ERAP1, you could get two different scenarios when you have an abnormal trimming, abnormal peptide, or abnormal antigen, and an abnormal immune response. Or the other possibility is already the B27 can misfold and accumulate, causing the excess inflammation. Maybe if the antigen which is sitting on it is also abnormal, you get more inflammation. So these are the two big possibilities, and it looks like these two genes are related and interact functionally. I'll skip through these. So what we have done is we have actually identified that if we introduce these different variants of ERAP1 into cells, we can actually induce the misfolding of these cells, and we have already published these results, which shows that there is significant increase in inflammation because of an interaction of different ERAP variants or the variants associated with ankylosing spondylitis and HLA-B27. So I'll wrap it up with this figure here. We already talked about HLA-B27, the possibility of misfolding. If there is misfolding, and it could be because of the interaction with the second most important gene, ERAP1, it can trigger the release of interleukin-23. You might be familiar with interleukin-23, but definitely you'll be familiar with TNF. Tumor necrosis factor is a molecule produced by cells which is pro-inflammatory. IL-23 or interleukin-23 is a similar molecule produced by the cells which is also pro-inflammatory, and that can be released by this HLA-B27. Now IL-23, again, you saw in the plot, the CN tower plot, that IL-23 is associated with ankylosing spondylitis. So we have a situation where B27 misfolds, causes unfolded protein response, releases the inflammatory cytokines. These cytokines interact with the receptors on again immune cells. That in turn results in release of other pro-inflammatory cytokines or interleukin-17. That interacts again and finally results in the production of tumor necrosis factor. And we know that treatment options are available now, which target tumor necrosis factor, which are the 5 agents available in the market today in Canada. Things which are coming up with Dr. Inman will be alluding to shortly are agents which target interleukin-23, very interesting results, and also interleukin-17. So there are several new options available for treatment of ankylosing spondylitis, which either are in the testing phase or almost there in the market currently. Okay, I think I'll stop there, and I'll welcome Dr. Inman to continue discussion regarding the treatment options.